Okay, folks, so we talked a little bit about temperature and how it affected the growth of bacteria. Now let's look at pH. All right. We know that temperature had an effect on enzymes. It could denature enzymes, and so it makes sense that it could also have an effect on growth. pH in the same boat, okay? We know pH had an effect on enzymes. There was a sweet spot when it came to pH and the activity of enzymes. And it's also the case that pH could be too low or too high for the proper shape of those enzymes. So it's going to affect growth, all right? Now, we could have a graph. I used a different kind of graph here, but we could easily have a graph like the temperature graph we saw before with the inverted U-shaped curves. But this just shows it a little differently. We have our pH scale from 0 to 14, and we have a number of different categories we could place organisms in when it comes to their preference when it comes to pH, okay? So most of the bacteria we look at in the laboratory, and most of the bacteria we worry about, frankly, when it comes to pathogens that infect humans, are actually neutrophiles. Okay, so we're talking organisms that are basically in the same range that our systems operate in, and so it makes sense that they would infect us, right? That these kinds of pathogens would be the ones we need to worry about. But again, there's going to be variations, so any one organism could kind of overlap in these categories. But the actual terms make sense, right? Just like thermophile made sense for an organism that was liking heat, right? Here we have acidophile, likes the acid environment, right? Likes low pHs. Alkalophile, these are organisms that are going to like it a little more basic, right? Higher pHs. And then, of course, we have our extremes, right? On either end of the spectrum, okay? Now, just to give you a couple of examples, neutrophiles, there would be, lots of organisms we could talk about there. Most of the organisms we grow in lab are in a pH 7 broth or agar plate, and they grow great, all right? But there are some that grow only on, let's say, fruit in your refrigerator, despite the fact that they are limited in growth because it's cold there, right? They're probably not going to like it at a colder temperature if they're a mesophile, right? Which is why we put things in the fridge. But some organisms will grow on your fruit nonetheless, right? If you pull out a peach or pull out some kind of fruit in the refrigerator and there's something growing on it, just fine. Those are more than likely not only they could be psychrophiles because it's cold, but they could also be growing on the fruit there because of the citric acid, right? So you might, in fact, on your peach in the fridge, have a combination of both, an acidophile, and maybe it's a psychrophile too. We ran into an issue some years ago with listeria. This organism was on the outside of cantaloupe. It had gotten contaminated where they actually harvested the cantaloupe. And of course, we don't eat the outside of cantaloupe, but when we cut through the rind, through the outside of the cantaloupe, we would drag the organism into the flesh that we do eat. Okay? Cut half a cantaloupe, eat some of that, stash it back in the fridge. Well, these were not only psychrophiles, but acidophiles that just kept on growing. Okay? And they caused some problems, right? Because these are pathogens. Now, example on the other end of the spectrum, in the alkalophile group, we have a couple of different organisms that, at least one of which I'm sure you've heard of, and that's Vibrio cholerae. Causes cholera. There's Alcaligenes faecalis, which you may not have heard, but right in the word it says Alcaligenes. The first part of the word sounds like alkalo, Alcaligenes, and it's in fact very happy at a little bit more basic environment. Okay, another environmental effect on environmental pressure that can have an effect on bacterial growth would be osmotic pressure. Osmotic pressure has to do with solutes in the fluids around cells, okay? Remember, osmosis has to do with water going in and out of membranes, and the effects of solute concentrations, well, if it's really concentrated around a cell, we could see where water would leave the cell, right? But and typically that would be really disadvantageous for a cell. It would probably kill the cell if you dried out a cell too much. But some organisms are quite happy in higher concentrations of solutes, like, for example, salt. Okay, so the pressures of high solute concentrations will kill a lot of cells, but not all. We call some of these cells osmophiles if they like that kind of environment, and a perfect example would be those halophiles that we've been talking about in lab, like those cells that grow on an MSA plate. Okay. So where might you find halophiles? Well, when it comes to close to home, like your own body, it would be skin. Okay, there's certain parts of the skin that will have more halophiles than others because it's saltier in some places than others. Okay, but generally it's a dry and a salty environment on the skin. But you'll see variation as you go around the body and you find different concentrations of that salt. But that would be a classic example of halophile. Another example I think of, this is a professor, I don't know if he's still over there, but he was a geologist at BU, at Binghamton University, and he was studying salt inclusions from ice samples that are, thousands of years old, but as the ice formed, the fluid concentrated to the point where there are these little salt inclusions, and what he claimed was that he could isolate bacteria from these salt inclusions. Now, this is controversial because, you have to show that you have a nice, clean environment and that you're actually isolating these organisms from the sample and it's not just some kind of contamination, but this is what he was looking at. We see these salt inclusions out in the environment. Here we have the Great Salt Lake. Definitely you'd find halophiles there. The Dead Sea, absolutely an environment we would find these halophiles. All right, so we've got pH, we've got salt levels. What about radiation? Radiation can have an effect on growth. Usually a negative effect on growth. UV light will form mutations within DNA, and this can curb growth. This can affect growth of bacteria. Ionizing radiation can break hydrogen bonds, so this can do damage to both the DNA and proteins. And radiation also oxidizes compounds so that they're not working any longer after that occurs. So this really explains why it is sometimes we'll use UV light, we'll use radiation to sterilize instruments. If instruments are, let's say, a plastic material, well, we can't put that through the autoclave, right? The autoclave would kill everything there, but it also melt the instrument. But if we have plastic materials, we can treat them with chemicals, or we can give them a dose of radiation to make sure they're clean and sterile before we use them in a place where we don't want to introduce contamination. All right, everything we've talked about thus far, we started with oxygen, temperature, pH, salt levels, radiation. These are all abiotic factors. They're not living, but they are variables that do impact growth. This last slide introduces the effect of other organisms. The fact that organisms, other species, can affect the growth of another species. Okay? Symbiotic associations exist. Here are three of them. Okay? you could have a mutual relationship, right? If the feeling is mutual, well, you feel, you both feel the same way. You're getting along, right? Well, in the case of an interaction between two organisms, like organism A and organism B here, it may be that the existence of the other organism is beneficial in both directions, okay? So organism A benefits from the existence of organism B and vice versa. Maybe some of the products of organism A, organism B uses. Maybe some of the effects of organism B being around affects organism A in a positive way. So it's a plus-plus relationship. Commensalism would be a situation where we have a kind of a plus zero effect. Okay, one organism definitely benefits from the existence of the other, but the other couldn't care, right? So we have this one-way relationship where one organism definitely benefits, but the other one isn't detrimentally affected, just could care one way or the other. Another symbiotic relationship we could talk about is parasitism. Okay. Now another way of thinking about parasitism would be predation. Okay. So if we think about a predator, right? We can just think about furry animals right now. A fox and rabbits, right? So fox are going to catch rabbits. They're going to the rabbits' population is going to be detrimentally affected by that. Okay. So this is a negative effect on one organism. But the other organism is definitely benefited by the fact the rabbits are around, right? So there's a predator. That would be the fox, right? Well, now let's put this in terms of parasitism. It's very much similar, okay? We have an organism that is negatively affected by a parasite. If I'm infected with an organism, a virus or a bacterium, then I'm going to be negatively affected by that. But the bacterium that's infecting me is going to be positively affected. So it's very much a predator-prey relationship, but we think of it as a parasite in that case. Okay, finally we have synergism and antagonism. A 0-0 effect where the two organisms could care less that the other's around, okay? And we have antagonism, where they both lose in the relationship. Another way of thinking of antagonism would be competition. Okay? As long as you are putting forth the energy to compete with another organism and the other one's doing the same, then there's a loss-loss relationship going on. Okay? So there's definitely a combination of both abiotic and biotic factors that will dictate the success of any population as that population grows in size. Okay? All right, next time we're going to launch into some microbial ecology, and we'll leave it at that for now.